All right, so let's go over chapter 6. There's a lot of stuff going on in this chapter, but I will try to not make this video too long. All right, let's get this up here. So muscular system. So I started off by just going over the idea about what is common amongst all the three types of muscle, right? So it's excitable, which means that it will respond to electrical stimulation. And it's contractile, so it shortens. So I know that's kind of a weird thing to think of because the next thing is that it's elastic. Usually when we're using elastics, you're pulling it outwards and it bounces back. But in this case, as we went over with the sarcomere, what happens with muscle is that it actually gets smaller. It contracts. And then when those myosin head groups release the actin, which I can tell you now because we already went over it, it's elastic in that it will come back out again, right? So remember, muscle contraction, the sarcomeres are getting smaller. And then the elasticity allows for the muscle to recoil back to its original length. And then the other thing about muscle is that you can extend it to a certain point because it is elastic, it will come back. But usually we don't want to extend our muscles too far because that's actually damaging to them. So if you pull a muscle, all right? So the functions of muscle, right? So produce or resist movement, and I'll go over the muscle pairs again, generate heat through shivering. Storage, that's going to become really important when we do a gastrointestinal tract, and that we have sphincters, which are smooth muscle. I know this chapter we're really concentrating on skeletal muscle, but sphincters will help store things or withhold them. And we'll come across those in many other places. And the movement of substances. So again, coming back to the GI tract, or that would be peristalsis. But we also have the constriction and dilation of vessels, so blood vessels, but also of conduits for air, so respiratory system, all right? We're going to be constricting and dilating those passageways, and that is also all smooth muscle, all right? Now, here with the muscle, there's two ends, right? The tendons are joining the muscle to bone, so the side that is attaching to the stationary bone is the origin, and the insertion is very often across a joint, all right, but attaching to the motile bone, all right? And during a contraction, like it says here, the insertion is pulled towards the origin. And I'm giving this picture here. It was actually a pretty good one, but it's kind of a weird muscle to be thinking of, all right? But it is indeed the case that the insertion is pulled towards the origin. And then we have our muscle pairs. Okay, so antagonistic pairs are ones that are completing a motion, but they're taking turns in their contraction and their relaxation. So one muscle is going to oppose or resist the motion of the other muscle. So, but they have to work together. So the good example would be the triceps and the biceps, right? So when one is contracting, when the biceps is contracting, the triceps must relax. But then to put the arm back to the starting position, the triceps has to contract and the biceps relaxes. So these are working together to do this full motion, but they are contracting in turn. So that would be antagonistic. Whereas synergistic muscles, they are contracting simultaneously. And usually synergistic muscles are supporting the prime muscle. So in this case, when you're bringing your weight upwards, so the biceps is contracting, there are a few other muscles that are also contracting to support that movement. So the brachioradialis and the biceps brachii. Oh, sorry, that's our brachialis. Oh my goodness, brachialis. So the brachialis and the brachioradialis are synergistic with the biceps brachii, okay? And here in this one, they have to, you can see this picture, they cut away the biceps so you can see where the other synergist is. So these three muscles are working together to bring this weight upwards, okay? All right. Then I started talking about where we get muscle, and specifically skeletal muscle, because that's what we're talking about most in this chapter. So what happens here is that these are very long muscle cells or muscle fibers, all right? Myofibers. They're all the same thing. So if you Google this, these are all the same thing: muscle cells, muscle fiber, myofiber. Okay. I think I stressed at one point that if you see the term myo or sarco, that is referring to muscle. Right, so these I always use the sartorius, and if I teach A&P, this is usually a muscle that I will label because it's so easily spotted. But you can see how long this muscle is, and there are cells here that are the same length as this muscle, right? So going from the hip to the knee. All right, and with that, the way these cells form is that they start off as these little myoblasts, and they fuse, and they keep the nuclei, all right? These are long multinucleated cells, and they fuse, and then as development moves forward, we get the full muscle cell. And remember why we need all those nuclei, right? Because that's where the messages are, or the DNA that makes the proteins that are required for the muscle. And it's much more efficient to have these cells all through the muscle, not just imagine if your nucleus was way up here, how it would take forever for proteins to get down here. And we'll talk a little bit about that when we do the nervous system, because that's the other place we come across these really long cells, but they only have one nucleus, all right? And how that's dealt with. Now, with a muscle, we can't be doing that because they're so active. So this is what's going on here, okay? Now, and then I went over the diagrams, and if you missed class that day, I'm sorry, but it's much easier to walk you through this as I draw it. And indeed, I did draw this in class. Here, I'm just going to go through this, and you're going to have to try and see if you can grab somebody's notes of my drawing. But basically, muscle is made up of these, not only these long cells, but layers and layers of connective tissue, and that's because we need to support the muscle. So the very outermost layer of connective tissue is called fascia. And I'm going to qualify this because your textbook calls all of these layers fascia, all right? But I want to teach it to you properly. Not that that's wrong, technically, but really only the outermost layer is the fascia. And then the other ones are given these specific names because it tells you exactly where you're at. So that's what we're doing here, all right? So if you try to look up these terms in your textbook, you might not find them, all right? So this is going to be in a standard A&P text, all right? Okay, so the outermost layer is the fascia, and you can see that the fascia is covering many muscles, and it's sort of underlying the layer of the skin. So there's a hypodermis, remember your fatty layer, and then you're going to start getting your muscle. So around all of those big muscle groups, or groups of muscle that are working together, are these fascia. And then when we get into a muscle belly proper, so one of these muscles, around that is a sheath of connective tissue called the epimysium. And this one is the one that's continuous with the tendon, all right? This one here, not continuous with tendon. This one here, continuous with tendon, all right? And it's surrounding the muscle belly, all right? So you can imagine, just say this is one part of the biceps. Now, under that, you're going to have these chunks of muscle cells called the fascicles, all right? And these fascicles, so if you're eating a steak and after it's cooked, you can see the fascicles. You can see those little clusters of muscle cells after it's cooked. The reason you can see it after it's cooked is because the connective tissue that's surrounding that fascicle, which is called the perimysium, has been broken down through the act of cooking. Now, inside these fascicles are your muscle fibers, your actual muscle cells, all right? So that's what's coming out here. And each of those muscle cells is surrounded by another layer of connective tissue, which is called the endomysium, all right? So that is surrounding the sarcolemma, or the plasma membrane of the muscle fiber. Okay, so we have that connective tissue. So and the reason we need all those layers of connective tissue is to help these things stay together, because the whole cells are contracting because there's this muscle. So we need that connective tissue to help preserve the muscle intactness, if that's a word. Yeah, probably not. But anyways, try and keep the muscle intact. All right. And again, you can see I've tried to walk you through this if you were in a class on page 2 and 3 of my notes, okay? Now, within the muscle cell, we have bundles and bundles and bundles of proteins, and those bundles are called myofibrils, okay? So these myofibrils, you can see here, if you cut it in cross-section, you can see there's stacks and stacks of these, cylindrical structures of of protein. And they're all grouped together there, and they're called myofibrils, and they come in two flavors, all right? And those flavors are called myofibers. So we have thick ones and we have thin ones. The thick ones are made up of a protein called myosin, and the thin ones are made up of a protein called actin. Okay, so that's what we're looking at here. So we have thick and thin. And these things are arranged in repeating patterns called sarcomeres. And it's that repeating pattern that causes these lines to be visualized by microscopy on these muscle fibers. Okay, so these lines are called the striations. Okay, if you go back to my muscle chart that I gave you, oh, what chapter was that? I think it was chapter 5, comparing all the muscles. So the striations, those are because of the sarcomeres, all right? So that repeating sort of system in which the thick and thin filaments are arranged. Okay, now, if we look at the arrangement of these filaments in the sarcomere, and I'll walk you through this again. I started with the M line. So the M line here is in the middle of the sarcomere, and it's holding the myosin. So M, middle, myosin. Then on the outside, and then I drew the myosin. On the outside, we have the Z disc, or the Z band, right? Sometimes, or the Z line, and sometimes it's called. But if you see Z, that's going to be the outside of the sarcomere, the outer edges. And here is where our thin filaments are being held. And you can see that they sort of mesh with each other. Okay, so the actin and the myosin, there's going to be some overlap here. And this is what we need for our contraction. Now, any place where you have myosin is referred to as the A band. Okay, any place you have myosin. And any place where you have only myosin is the H zone. So you notice that the difference between the A band and the H zone is that the A band includes this overlap with actin. And then any place you only have actin is called the I band. Okay, so that's here. And you can see that one I band is going to feed into the next sarcomere, because there's going to be another one over here, and then there's going to be another one over here. Okay? And then here, if you look at this guy as a bundle in 3 dimensions, you can see that these filaments are all sort of arranged so that they can slide past each other, all right? And what ends up happening, and think about this, when this sarcomere gets smaller, the A band will stay the same size, okay? Because it's not the myosin that's shrinking, and it's not the actin that's shrinking, it's just sliding past each other. So the H zone actually gets smaller. Okay, so hopefully that makes some sense to you. Now, how do we get this thing to contract? All right, so then I brought us back out again. And remember, they respond to electrical stimulation, and that electrical stimulation comes from neuron. So the neurons, they extend out and they interact with the muscle cells at these motor end plates. They're sort of like these bulbs, and there's going to be terminal end bulbs, each of these little bulb. And in class, I drew a bulb, and then below it, I drew part of a muscle cell. And what will happen is that the neuron, which has an electrical charge running down the axon, and we'll go over that later in the semester, it comes down here, and there's a space here. So it can't, the electrical stimulation does not jump from the neuron to the muscle cell. What ends up happening is that the neuron releases neurotransmitter, and that then binds to receptors on the muscle cell that then starts this whole electrical transmission again. So the neurotransmitter that's being released is called acetylcholine, all right, and it's released. And we'll go over how this works later on in the semester. But suffice to say right now, we've got release of neurotransmitter, and it interacts with this muscle cell membrane, and then that sets up the electrical impulse or the charge on the muscle cell. Now, still we have to get that charge associated with some motion in the sarcomere. So how does that happen? Well, in the muscle cell membrane, what it does is it makes these holes or tubes that extend literally, you know, they're lined, these tubes, these holes. This is still the cell membrane. It goes deep into the cell in these tube structures, and these tube structures are called T-tubules. So the electrical impulse travels down that T-tubule, and it interacts with a structure within the muscle cell called the sarcoplasmic reticulum. The sarcoplasmic reticulum now is basically endoplasmic reticulum, but it's smooth endoplasmic reticulum, so it's not rough. Remember, rough endoplasmic reticulum we use for making protein because it's coated with ribosomes. But remember, the smooth endoplasmic reticulum had a few functions, and one of them was storage. And the sarcoplasmic reticulum, what it stores is calcium. And this calcium is released once the electrical charge travels down the T-tubule and interacts with the ends here of these sac-like smooth ER, or sarcoplasmic reticulum, and that calcium comes in, and it then starts the contraction and relaxation cycle, or the cross-bridge cycle, however you want to read it off Google, and I'm going to go over that in a sec. But just finishing off the structure here. So here you'll see this T-tubule and the sarcoplasmic reticulum is perfectly aligned over a sarcomere, all right? And one T-tubule and two ends of the sarcoplasmic reticulum are referred to by this term called the triad, okay? And you can see that it's right here, as I wrote it here, it's in the region of the A-band, I-band junction. So it's sort of right over a sarcomere. So you can see as many sarcomeres as you have also got it bordered by T-tubules and triads, all right? So that calcium gets released. So what does that do? Well, let's think about it. Okay, so here's another look at the T-tubule. I just like this picture. That's why I added this slide, because I think it, you can sort of see that on the surface of the muscle cell, there's these, they look like pores. They're not really pores, but they're holes that go, continuation of the membrane in these T-tubules. Okay, and then I showed this video of sarcomere contraction, which you can watch again. But notice, here's a relaxed sarcomere, here's a contracted sarcomere, and you can notice that the A band has stayed the same size, but notice that the H zone has gotten smaller. Okay, and the other one that's going to get smaller, I don't refer as much to this one because it's between two sarcomeres, but the I band as well. But the H zone definitely is getting smaller, and you can see that here. If you look at this using the microscopy, you can actually see using the EM microscopy that is indeed what happens. Okay. All right. Now, so remember now, in a relaxed muscle, the actin and the myosin are not interacting, and in a contract muscle, the actin and myosin interact. They slide past each other. So clearly something has to change. So that calcium is doing something. So what is it doing? So let's review the structure, and then we'll get back into that. So the thin filaments are made of these little balls of actin, all right? They make, these called G-actin individually, and together they make F-actin or fibrous actin. And there's two of those F-actin molecules that are wrapped around each other, and they're stabilized by another protein called nebulin, all right? I don't really talk about it in this class other than the fact that it's holding these two chains together. Now, myosin, on the other hand, one molecule would be like two tail groups sort of wound around each other with these little head groups on the other side, on one side. And these guys are motor proteins, so they use ATP and they walk, right? So we'll go over again how that works, all right? Now, if you look at the M line, it's holding bundles of these thick filaments together, all right? And here you can see a little bit better this hinge region. This diagram is a little bit better, okay, in terms of what the actual structure might look like. So what happens when there is no calcium? So when the muscle is relaxed, there are these other two proteins that cover up the myosin binding site on actin, and they are called troponin and tropomyosin. Troponin is bound to tropomyosin, and this is the protein that binds to calcium, or that the calcium binds when it's released into the sarcomere. And the tropomyosin is covering up the myosin binding sites on the actin. So what happens is that the calcium binds to the troponin, and these are positively charged ions. That positive charge changes the shape of the troponin protein, which then in turn changes the shape of the tropomyosin protein, and these guys move out of the way, and then actin and myosin can interact. Okay, so here we have the naked actin fibers or F-actin, you can see represented by these black dots where the myosin would be able to interact with it. And when the regulatory proteins, troponin and tropomyosin, are not associated with calcium, you can see that they're nicely covering this up. But once we add calcium, what's going to happen is that the tropomyosin is going to slide out of the way, and the myosin binding sites will be exposed on the actin or the myosin. All right, so then these little head groups can reach up or down. I mean, I keep saying up. I shouldn't do that. That's why this picture I've purposely done down. It reaches out and binds to the binding site on the actin. Okay, so now what? Okay, well, they're bound. That's still not having this thing contract, all right? So you can see the difference here. Here we're covered up and relaxed, and these little head groups are down here. And now when the calcium's there, now these guys have reached up. But how are these guys walking? Well, I already said that they're motor proteins, and if you were in class, you know exactly what we're talking about. But just remember, the electrical impulse has traveled down the T-tubule. Calcium has been released, and it is binding to the troponin, which moves the tropomyosin out of the way. So clearly, one of the molecules that we need for contraction is calcium, all right? Now, the other one that we need is ATP, all right? And I'm going to walk you through this picture again. In class, I broke it down. If you missed that class, again, that's unfortunate. But anyways, I digress. So what ends up happening is that the myosin head groups, they are energized with ATP. That ATP splits, and it remains bound to the head group. So we have ADP and phosphate, and that splitting causes the head group to cock or reach out, and then it can then touch the actin. Okay, it reaches up and touches. And when it does so, the phosphate group falls off. When the phosphate group falls off, we have very tight binding of the myosin on the actin. Now we still have the ADP bound. So what happens now is that the ADP comes off, and when it does, it causes the hinge region to bend, and the head groups bend and pushes, or pulls, I should say, pulls the actin, all right, thereby shortening the sarcomere. But now we have to allow detachment, and the way that this detaches is when a new ATP molecule binds, and it will then detach, and then we start all over again. Okay, so you can see this broken down in my notes. What page are we on? Broken down, starting on step 1, page 7 of my notes. Okay, so we have the energization of the myosin head group, the cross bridge formation. And it's always hard to draw that first step because there's two things that are happening. The cocking of the head group and then the touching of the myosin on the actin. So then step 2, the cross bridge formation, the power stroke starts on page 8, and also on page 8, detachment of myosin from the actin, all right? And then please watch these 2 videos again. Since this is a cycle, and I've had students in past go on and Google this, all right? And they get really angry with me because they go, oh, no, step 1 is, you know, the binding of ATP, or, you know, the cross bridge, or whatever it is. Remember, it's a cycle. So different professors that you might listen to online might break this down a little bit differently. So I kind of looked for videos that went through it the way your textbook does and the way I've presented it in class, all right? But understand I'm also presenting it this way because it's sort of how it's done in your book, right? It's a cycle. So you can start anywhere, all right? To me, it's just most logical to start where the head group is binding onto the actin, all right? But don't let that confuse you. The steps are still the steps, and they might be massaged a little bit differently on how it works. I'll be trying how you read, but the way it works is still the same, all right? So don't get all panicky, all right? When in doubt, just go with what your professor is telling you, because that's how I'm going to test you. I know that sounds, you know, like a dictatorship, but I'm sorry, I have to choose one of the ways of presenting this information to you. So let's just agree on this one, all right? I know that sounds weird, but after teaching this for 8 years, different students will Google different things. Google is great. It's great. It helps. But just remember, it's a cycle, so you can start anywhere. All right. Okay. I digress. So here we go again. So first we have ATP hydrolyzed. So that's your first molecule of ATP that's needed. The splitting of that causes the cocking and the reaching up and touching of the actin by the myosin. When we lose that phosphate, that group, that head group binds tightly to the actin. Then we lose our ADP, and that allows for the myosin head group to bend or rotate, however you want to describe it, but that's the power stroke. And that causes the shortening of the sarcomere. And then we need a second molecule of ATP to come in and rescue this and keep it going. And it allows for the detachment so that we can reset and do this again. Okay? All righty. So we need at least 2 molecules of ATP for each motion of this head group, each head group, all right? If you watch the videos, you'll see that these head groups are literally walking along. Now, where do we also need ATP? We also need ATP to pump calcium back into the sarcoplasmic reticulum. And understand this is happening all the time. It's not like a switch. It's always being pumped back in. But when we have an action potential or electricity flowing across the surface of the muscle cell, and it's coming down to the T-tubule and releasing calcium, that is going to outweigh this, all right? So what will happen, eventually the neural stimulation will stop, because you know when muscles are not constantly contracted. When this happens, this is going to take over, and this calcium is going to be mostly then pumped back in until we lose calcium in the sarcomere. And what will happen then is that the troponin tropomyosin will cover back these the binding site for myosin on actin, and we'll have a muscle relaxation. Okay, so hopefully that makes some sense to you. Then I went into this whole thing, and man, this looks pixely, but it's hard to find a good zombie picture. But but definitely better than pictures of rigor mortis. Don't know what I mean? Google rigor mortis and look at images. So I thought this was a little bit more benign. I wanted to have an image to put up while I talk to you about this. So rigor mortis, you know, that's the stiffening of muscles after death. Now, why does that happen? Well, you're no longer breathing, you're no longer respiring. So why do we breathe? Well, we need to provide oxygen to make ATP in our cells, right? So if we go back here, you know, if you don't have ATP, right, this guy will not let go, right? So the myosin head groups will remain attached to the actin, and the muscle will remain contracted. So without any ATP, that's why these muscles remain contracted. On top of that, we don't have ATP to do this, to pump this calcium against its concentration gradient back into the sarcoplasmic reticulum. So the sarcomere remains flooded with calcium, and the myosin binding site on actin is exposed. So this is why after death, we're not making ATP. We've still got the flooding of the calcium, and we can't release the head group from the actin, all right? So that's why that happens. And then eventually things start to break down, as we know, and these proteins will break down, and then the muscle will then relax. So this gives one of the ways of timing death, right? So I know that sounds gruesome, but that's part of it. So we'll explain that in this class. Now, clearly, we need lots of ATP to power the muscle. So where does this come from? Well, we have 4 sources of ATP, right? And understand they're all present. It's not like, you know, one is used up and then the next one gets clicked on. But I'm going through it logically in which sources are going to be the prime draws as muscle is working, right? So the first source is clearly ready-made ATP, right? But the muscle cells only store up enough to maybe power about 3 seconds of work. But then in the second place is that some of that ATP that has been converted to ADP can be recycled by drawing phosphate off of this other protein called creatine phosphate. All right. So creatine phosphate, when a muscle is relaxed, creatine is becoming creatine phosphate. It holds on to phosphate groups so that the muscle can recycle ATP quickly. So then when there's more ADP around, it will take that phosphate off the creatine phosphate, make a new molecule of ATP, and then we're left with creatine. All right. So that's the second source, and that lasts 12 seconds. Okay, that was my trivia that I looked up there. Then we've got to start making ATP, right? And not that this is not happening, but then we're going to rely on made ATP. In the first place, we make a little bit of ATP is by glycolysis. And sometimes our muscles are not well oxygenated because we're working out, we're using up oxygen too quickly. And what ends up happening is that we sort of start relying on this first step, glycolysis, which is anaerobic. It doesn't require oxygen, all right? You're making a little bit of ATP, but if you're not having enough oxygen, what will happen is that the body will try to keep making ATP this way, and a byproduct of that is going to be lactic acid, which is one of the reasons we can get muscle cramps. Later on, when you're resting, that lactic acid is going to be shipped out into the blood, and then the liver is going to purge you of it, and you're going to make sugar again. But basically, that's what the body does. But if you're in well oxygenated muscle, what will happen then, of course, is that you're going to use those mitochondria, and you're going to make lots of ATP aerobically, and that's going to last you for hours, right? Until your muscle just fatigues, all right? Okay, so the different fiber types. Oh, yes. So this is just sort of a pictorial way of thinking about how we're using ATP. So first, ready-made ATP, recycling ATP with phosphocreatine, glycolysis, and of course, if we have a well-oxygenated muscle, we'll switch to aerobic metabolism from the anaerobic. All right. Now the fiber types. So you know that there's different colors of meat, and because of that, we have different types of fibers, and they're different. We use them for different things. So the red muscle, okay, so that would be the dark meat, chicken legs, right? Chickens use their legs to run around. These are called slow twitch fibers. They're very low in glycogen, so there's not a lot of sugar stored up there, but we have a lot of myoglobin. And in class, if you missed what I did to introduce this, is remind you about that old protein that you talk about most biology classes, and indeed we will as well, hemoglobin. And I was asking you, what does hemoglobin do? And somebody remembered that hemoglobin carries oxygen in the blood. Well, muscle cells have a way of holding on to oxygen, and that is this protein called myoglobin, and it resides in the muscle cell and it stores up oxygen. Why does it store up oxygen? Well, because we just said that the best way to make lots of ATP is using aerobic or with oxygen metabolism, right? So muscle cells, these red muscle fibers, have this myoglobin protein that holds on to oxygen, and that's really handy, right? Because we need to store up oxygen. So even if you are not getting enough oxygen, eventually you're going to use up the oxygen on your myoglobin, but it is there to help make ATP. And consequently, we also have more mitochondria. We have more storage of energy, right? And a greater blood supply. So why is this muscle red? Well, we've got a greater blood supply. Blood oxygenated is red, right? And myoglobin, if you think back, what is in the globin there to hold on the oxygen? Well, it's iron. And what happens to iron when it's oxygenated? It looks rusty. It's red, right? So that's what's happening here. So myoglobin holds iron, okay, and it helps add to that reddish color, all right? Now, the metabolism here is aerobic. Now, the white muscle fibers, those are called fast twitch. It's sort of the opposite of red, right? It's got lots of glycogen in it, so we're able to use a lot of sugar quickly. Less myoglobin, clearly. We don't have that iron in there making it red. Less of a blood supply, and we're not using oxygen as efficiently here, so we have less mitochondria. So here, this muscle is can be powered by anaerobic metabolism, all right? So that's like the chicken breast, right? Okay, so let's think about this practically. So slow twitch fibers are the ones that you're going to want to use for endurance activity. They contract slowly, but they're really good at for long, sustained activities, right? So aerobic training is endurance training. And the body, what it's learning to do is take in oxygen more efficiently. It's going to build the capillary levels, build myoglobin and mitochondria in those muscles, all right? So, but look at this guy's muscles compared to this guy's, right? It's a different type of athleticism. Now, this guy has very thick muscles. So what he's doing here is using the fast twitch fibers. These are ones that are trained in strength training. They contract quickly. They're very strong, used for brief, high-intensity activities. So we don't need lots of ATP production here, but we need enough for quick bursts of activity. So what's happening here when you're strength training is that the muscles are learning to work harder and quicker. You're not making more cells, all right? You're not increasing the cell number, but you're making those cells get bigger. So you're going to be building myofibrils, right? And you're going to be gaining in glycogen and creating phosphate for quick energy bursts, all right? So that's sort of the difference. I mean, these guys are both athletes, but their muscles look very different, clearly, right? Then the movements. So some of your muscle contractions result in movement, right? Those are the ones you think of the most. So concentric contraction or an eccentric contraction by raising or lowering a buck. Those are called isotonic. If there's a movement involved here, isotonic. But there's lots of contractions that don't result in any movement at all. So think about the muscles that are contracting to keep your head straight or keep your back straight while you're sitting in your chair. In this case, holding the book outright, right? So this is called an isometric contraction, right? So there's no movement involved. And I will ask you a question like this on the midterm. So describing a situation, and you'll have to define whether it's an isotonic versus an isometric contraction. Okay, now the motor unit. The motor unit is basically a motor neuron or a neuron that's supplying a muscle cell and all of the muscle fibers that it contracts. So one neuron can reach out and touch more than one fiber, all right? Now, here you can see there's 3 motor neurons here, and you can see that these are touching different muscle fibers, all right? So there's a whole bunch. So one motor unit is going to be this motor neuron and all of the fibers that it's contracting. Now, the strength of the tension that's produced there, the mechanical force resulting from this muscle contraction, is going to depend on a few things. The number and the size of the muscle fibers in the motor unit, okay? So how many, because they're going to be all contracting at once, right? This is all or none, okay? The number of motor units active in any muscle at any one time, so how many neurons are stimulated, and how frequently not only the neurons are stimulated, but also how frequently, consequently, the muscle fibers are stimulated, okay? So then you think about this. We have different types of units. We have high precision ones, okay? In this case, the high precision ones, there's fewer fibers, but we are allowed for very fine, delicate movements. So think about eye movements. So those would be high precision motor units. And then low precision ones, that could be a neuron innervating, thousands of fibers. Now these ones are not going to be fine movements, but powerful ones, all right? So strength, so like your thigh muscles, for example. So what we can do is that we can actually measure the electricity flowing through these fibers. And we're going to come back to this later on because we're going to look at the same kind of thing with a neuron, and we're really going to break it down how this works. But what we can do is we can measure the electricity flowing through the neuron. We can also measure the electricity flowing through the muscle fiber, and then we can also see the movement or the contraction. The movement is called a twitch, a muscle twitch, okay? So if we put in our sensor in the neuron, that's where we're going to see the electrical activity first, okay? So this electricity, the flow of that is called an action potential, all right? Because it potentially will result in an action. That's where the name comes from, I guess. I mean, it would make sense. So here we have the electricity flowing through the neuron. And then, of course, afterwards, right, we have to have a release of our acetylcholine, our neurotransmitter, and it has to go through the synapse, and then it has to bind to receptors on the muscle cell. So the muscle cell action potential is going to be a little bit later, right? But that electricity has to flow across the fiber and then down into the T-tubules. So this all requires some time, right? We have the electricity through the neuron, then electricity through the muscle fiber, even before we get a contraction, right? So this period of time is called the latent period. Okay, so that's the delay to the contraction. So it's sort of the spreading of the electricity over the surface and down the T-tubules. Then the muscle is going to contract. So that's called the contraction phase. And then it's going to release, right? It's going to have to release, and that's then called the relaxation phase. All right, so the latent period is before we get a contraction. Contraction phase, like its namesake, is when we get contraction. And then after that, we got a relaxation phase. Now, one of these cycles is called a twitch. Okay, so one cycle of contraction and relaxation is called a muscle twitch. Okay, now if we look at this graph, so we've got one twitch, right? And then we have another twitch. And the thing to remember here is that either the sarcomere is going to contract or it's not. It's going to contract or it's not. And the amount that it contracts depends on how much calcium is available, right? So if you use up your calcium, you're done contracting, right? And each cell is either going to contract or it's not. But remember that muscle is not just one cell. It's a whole bunch of cells, okay? And overall, right, the amount of force that a muscle is going to contract with is going to depend on how frequently the fibers in that muscle are stimulated and how much time they're given to relax. Okay, so what can end up happening is that we can take all of these twitches and we can add them together. So the more frequently we have twitches, all right, and the less time that a muscle has to relax is going to summate or add together until we get a full force contraction, which is called tetanus. So all of these twitches are going to come together. And the number of them will increase, the amount of time in between will decrease, and we'll get full-on tetanus or full-on when the muscle fibers and the overall muscle force now remains in maximal strength, okay? So if we have tetanus, the muscle fiber is not given an opportunity to relax, all right? So hopefully that makes a bit of sense, okay? Now, tetanus, okay, is also an ailment, and that's produced by a virus, okay? A bacterial infection. What am I saying? A virus. It's a bacterial infection of the muscle, and the tetanus bacteria, they produce this toxin that causes the overstimulation of the muscle fiber. All right, and that's because it's affecting the neurons. All right, so this toxin is going to overstimulate the motor neurons, and it results in full-on tetanus of the muscle of the muscles, so rigidity of the muscle. Now, important thing to remember here is that your diaphragm, your breathing muscle, basically, we're going to talk about it later in the semester, is a muscle. And when that one is overstimulated and it's contracted, you can't relax. And this is why it's so dangerous, can die of suffocation because you can't breathe properly. So you're routinely given the tetanus vaccine against this bacterial infection. Even as an adult, I think it's every 12 years is the regimen. I always get it when I go for my physical. They'll ask me, when's the last time you got a tetanus? And it's usually so long I can't remember, so they give it to me anyways. But I think the regimen is every 12 years. That's the last I checked. But the thing is, that way, if I would get some sort of a puncture wound, right, which is where you can be exposed to these bacteria that produce this toxin, then I'm vaccinated, so I will be okay. All right, so that's sort of a prophylactic thing. That's why we get vaccinations. Okay, then what I did is I started comparing the muscle fiber types, and we're going to be talking about each of these quite a bit, right? So cardiac muscle fibers versus skeletal muscle fibers. You can go back to that chart. I always recommend that you print that chart out, comparing these things. It gives you sort of like the key things to remember. But remember, cardiac muscle and skeletal muscle have some similarities, all right? So they're similar in that they're both striated, and they each have the regular sarcomere structure. But they're different in other ways. So in this case, the cells are smaller, all right? Cardiac muscle cells are not those elongated, multinucleated cells. So these cells are smaller. They can be branched with single nucleus. And importantly, they are involuntary. You don't control the beating of your heart. Your heart controls itself, all right? So it's involuntary, and we're going to talk about how that works in great detail. But the other thing is that in their structure, they have these things called intercalated discs. So you can see them here as these dark bands. Now, these intercalated discs have two key proteins in there. One of them, they have those adhesion proteins, right, that really hold these cells together. And I think the heart is beating for your lifetime with great force. So it requires those extra adhesions there at those intercalated discs to keep those cells from breaking apart. The other really, really, really important thing is that there are gap junctions in there. So remember, gap junctions allow for the cytoplasm of one cell to flow into another one. So the ions can move through here. And this is in part how your heart works, is it doesn't require each of these cells to be innervated by a neuron. When one set contracts, and we'll talk about that in a sec, it will then send a signal to other cells to contract, all right? So they can directly stimulate neighboring cells through the gap junctions in these intercalated discs, all right? So intercalated discs have adhesion junctions and gap junctions, all right? And that is only in cardiac muscle fibers, these intercalated discs. Now, some of these cardiac muscle cells are called pacemaker cells, all right? So what they do is they set the rhythm. And I showed this video in class where you can see these cells contracting all on their own, all right? They have their own, they've got special, we don't talk about it in this class, but their resting membrane potential wanders. And because they're a little different. And but suffice to say that they set the pace for the rest of the heart muscle, which is why they're called pacemakers. And that's why when your heart isn't beating well on its own anymore, they give you an artificial pacemaker to set up that electrical connection. Okay, so these are sort of, they time the heartbeat, right? They contract on their own, and they can do so even without the influence of the brain. So think of they are influenced by the brain, and we'll go over how later on in the semester. But if someone gets a heart transplant, that they're connecting up the vessels, but they're not connecting the neurons. That comes later. But that heart can contract on its own and set its pace, all right? So the donor heart will do that already on its own. Okay, so the brain does influence it, and we'll talk about how later, all right? Okay, now smooth versus cardiac, all right? Now smooth muscle fibers are different. They're not striated. You don't have that sarcomere structure, all right? So like cardiac muscle fibers, they are involuntary. You don't control the flow of food through your gut, okay? Or the flow of food, of blood, I should say blood through your vessels. They are electrically linked to one another. They can be by gap junctions, so I'll sort of qualify that shortly. Some of them can have a pacemaker potential, but it's not quite like what you see with the heart, but it's a little different. We will talk about it, right? These ones are also under the control of the brain. They are influenced just like the heart ones. But unlike cardiac muscle, they don't have a sarcomere structure, and they do not have intercalated discs. Then what I ended up doing in class is talking about the structure, and I want to plant the seed. We're going to talk about it later. But smooth muscle is very often associated with rings, and these rings are going to be in different directions. So here, if you look at these, this would be an in-plane cut, and this is sort of wrapped around a tube, and I drew this in class, right? You have a lumen of some kind of space, whether that be what we use for breathing, so your trachea, and all that other great stuff, which we'll talk about later, or the middle of your digestive tract, or the middle of your blood vessel. You've got a layer of smooth muscle wrapped around that, and then you're going to have another layer longitudinal to that, so at right angles, right? And these guys will contract in different ways, but you can use it then or imagine how this can cause the constriction or the shortening of the tube, or not the shortening, the small decreasing diameter of the tube, or the dilation or the increasing diameter of that tube, okay? Now, when it comes down to the structure, we don't have sarcomeres, but we still have actin and myosin interacting with each other, okay? So actin is going to be held not by the Z-bands, but by these little dense bodies. And in between, we're going to have our myosin. And the myosin head groups are still going to walk along the actin. We're also going to have connective tissue connecting different smooth muscle cells so that this can contract as a unit, all right? Okay, I'm going to just look at my notes to make sure I'm telling you everything that I'm supposed to tell you, reminding you of everything. Okay. Now, here we still have the myosin head groups that are going to walk along. Now, in this case, these head groups are going to be on the full length. Notice that they're going in different directions. So this actin is going to be going this way, and this actin is going to be going that way. So what's going to happen here as a result is that the entire cell is going to shrink, all right? So we're not having the sliding thing. We have this meshwork. These guys are walking in opposite direction, are pulling the actin in opposite directions as they walk, and the whole cell is going to contract like this, like a little, like a pillow. All right, you're squeezing a pillow somehow. I don't know. You can use your own sort of idea about what's happening here, but it's different than a muscle cell, right? I mean, a skeletal muscle cell. All right. I mean, this muscle cell is also contracting. Skeletal muscle cell is also contracting, but not in, you know, in every direction. It's going to be decreasing in its length. But here you can see the smooth muscle cell is contracting in all directions. All right. So it's not quite the same kind of thing. All right. Now, when we have the neurons interacting with these muscles, there's different types. There's single unit smooth muscle and multi-unit. So what's happening here? And here we have our lumen, right? And here we've got this neuron that has these, you know, viscosities, we call them, right? So it's like there's these bulbs all along the length here where it's interacting with the muscle cell, and it's going to be releasing a neurotransmitter. Now, in these single unit smooth muscles, these ones are connected by gap junctions. So what will happen is that all of these fibers are going to contract in unison, okay? And this is where we need a lot of control and a coordinated movement, all right? So you can imagine if this is a lumen, you'd want this whole thing to contract or constrict together, right? Not, you know, constrict here and not over here. So here it would kind of make sense that we want all of this smooth muscle to contract together. Now, then we have multi-unit, where we really need to have certain muscle cells contract and not others. And these ones are not going to be electrically linked. So this would be like controlling your eye movements. So where these muscle fibers, these smooth muscle fibers will contract on their own, okay? So a little bit different on how this is working. Then we had this little discussion on muscle imbalances, so cramps and pulled muscles. So pulled muscles, you're stretched too far, right? So there's going to be some bleeding, right? That's at the ends if it's severe. It hurts. And that's because now we need to repair those proteins that hold those muscles together. Muscle cramp, that's involuntary, very painful contraction, all right? And it can be for a few reasons. And I list them in my notes. So electrolyte or ion imbalances, right? Inside versus outside. So this can happen from overuse. Dehydration, right? Not getting the right water balance, and the ions are not going to be in the right concentration. Inadequate blood supply. Okay, so low levels of oxygen. Remember, you're going to have less ATP, and you won't be able to have those myosin head groups let go. So that would be, you know, an involuntary muscle contraction. Now, muscle spasm is a little bit different. Single involuntary contractions, which are then released. So it's not generally the entire muscle. It's going to be some of the cells. It feels weird, not as painful, okay, but the reasons can be very similar. Then we have these two much more serious things. So one is called myasthenia gravis. This is an autoimmune disease targeting your muscle fibers, and specifically the neurotransmitter receptors. So it ends up being weak muscles. Usually noticed, I have it written here on the slide, by facial muscles first. And it can be treated, all right? Muscular dystrophy, this one is a genetic situation. So there's a loss of a protein called dystrophin. And what ends up happening is that with time, there's a lot of scar tissue that gets built up because the muscles kind of get damaged because the muscle loses its ability to control the amount of calcium that it has within it. And eventually the muscles are replaced by connective tissue and a weakening of the muscles with time, which results, unfortunately, in an early death of these patients, okay? And I think that's it for everything that I talked about. In my notes on page 15 and 16, you'll see 10 steps really in detail, which traces muscle contraction right from the electrical impulse to the relaxation. I don't get into too much detail with the relaxation, but it's basically the reverse. I really look over those notes. There's going to be a question on the midterm that's going to ask you to place the sequence, and it's not going to have all 10, but I want this to make sense to you so that you'll be able to piece it together, all right? I like those sequence questions because it tells me that you know what you're doing and you understand. All right, so that is the end of this little video. So hopefully you've been able to go through this and it's helped. I know a lot of you have been away, so hopefully that will help. All right, and as always, if you're confused, ask questions. All right, thanks so much.